So my talk today was about immune signatures in the tumor microenvironment, and for most people that keep up with the literature, they find it incredibly complex. There's hundreds or thousands of papers that talk about biomarkers or immune signatures for things like immunotherapy response. There's different types of immune signature. We group them into 4 different categories. First of all, the sources of antigen that can elicit a T-cell response, then the drivers of immunescape, then different patterns of immune infiltration and signaling, and then finally host factors. And so we tried to address this problem by doing a systematic review of the literature. We found over 700 papers that we reviewed in detail, and then we measured biomarkers from each of those papers, over 50 different biomarkers in a meta-analysis published last year. And from that, across 1,000 patients, we tried to understand which biomarkers could replicate, which ones didn't, to help understand the most important drivers of immunotherapy response. And from that work, we found the number of mutations, particularly the clonal tumor mutation burden in a patient's tumor, was critical for immunotherapy response. Also, expression of key chemokines, including CXCL9, had a very strong effect size in predicting checkpoint inhibitor response. CXCL9 recruits T cells to tumors as well as to tertiary lymphoid structures, so it's critical in achieving an effective immune response. Another chemokine, CXCL13, also was identified as very predictive of positive immunotherapy response. CXCL13 typically is known for helping form tertiary lymphoid structures, but also it's expressed in T cells directly, in CD8 T cells, and it can be a marker of CD8 T cells that react to tumor neoantigens, as well as the type of T cell that drives effective immune response. So those were our discoveries, and taking that forward, we're trying to combine them together, those different measures, and find ways, multivariable predictive ways, to make better biomarkers to identify patients that will respond to checkpoint inhibitor treatment.